Hello, my name is Carl, and in this video I'm going to briefly discuss the process of transcription in a prokaryote organism. Before we talk about transcription, let's briefly establish what a prokaryote is. So a prokaryote is an organism that has no nucleus, and mainly, especially for our purposes, this means bacteria. Bacteria have their DNA arranged in circular chromosomes, which is unlike eukaryotes, like us, who have linear chromosomes. In transcription, we're going to copy a segment of this DNA into a molecule of RNA. So you can see here our normal DNA in its anti-parallel strands with our 4 bases: guanine and cytosine, and adenine and thymine, except in the case of RNA, thymine will be swapped for uracil. We'll see this in a moment. The strand of DNA being read will be called the template strand, and its complementary strand will be called the coding strand. The template strand will be read in the 3' to 5' direction, and RNA will be transcribed in the 5' to 3' direction. The sequence of RNA will match up with the coding strand, which is why it's called the coding strand, except in the case of RNA, the thymines will be swapped out with uracil. So how does transcription happen? Well, we start with the initiation stage. You see here we have a segment of DNA where the green portion denotes the sequence to be transcribed, and then upstream or preceding it, we have the orange promoter region. At the +1 position, we denote where transcription will begin, and then we have a -35 consensus sequence and a -10 box consensus sequence. Consensus sequences are sets of commonly encountered nucleotides that contain considerable similarity. And in this instance, these sequences are going to act as promoters for the binding of our polymerase enzyme. Our transcribing enzyme is called RNA polymerase, which consists of a 5 subunit core enzyme that associates with a sigma factor to form a holoenzyme that will actually begin the transcription process. Our holoenzyme binds to the promoters with the active site of the enzyme over the +1 position and begins to unwind the DNA to form a transcription bubble. Free RNTPs in the cell form the building blocks of our RNA as they are bonded together by phosphodiester bonds. Our enzyme transcribes the sequence and the sigma factor dissociates. But how do we end this process? Repeating AU sequences can serve as pause sites for transcriptions where either a protein called the Rho factor can remove the RNA, or complementary sequences can form a termination hairpin that destabilizes and dissociates the RNA. Before we finish, let's speculate as to what a mutation in the promoter region might do. Here we have a 5' base pair insertion in the aminotransferase gene promoter region that would be used in the synthesis of phenylalanine. Our holoenzyme can no longer efficiently bind to the promoter region, which could result in decreased gene transcription, reduction in phenylalanine synthesis, and possibly disease or fitness reduction. That's it for this video. Thank you for watching.